I am acting division director within the Office of Manufacturing Quality, and which is in a sub-office within the Office of Compliance. My responsibility, or the responsibility of Office of Manufacturing Quality, or OMQ, is the enforcement of federal law related to adulteration of drug products. And that is going to be sort of the framework in which I build my presentation around. So when we speak of, or when the agency speaks of drug quality, drug quality cannot be tested in. The intent of, and I'll talk about the reason why in a moment, the intent is that it needs to be built in. It should not be random or accidental. It should be intentional, predictable, and consistent. And talk about more of that as we go along. And when we talk about, or when I talk about drug quality, I refer to the identity. If we're talking about drug products, the identity of the finished drug products. If we have, for example, fentanyl injection, is that indeed the fentanyl is in there. The strength corresponds to the label strength, plus or minus some tolerance. That it is purity, that it is pure in regards to certain specifications, and we can talk about other quality attributes. We can talk about fill volume. If we're talking about a batch, content uniformity. There are many things that that drug quality is measured against, but I'm just presenting some of the more well-known or and originally understood. All right. So as I indicated, that I'm going to use federal adulteration prohibitions as framework. So there's Section 501A1, it's prohibition against a drug containing any filthy, putrid, or decomposed substance. The language from 1938. 501A2A is prohibition against insanitary conditions. Section 501A2B is prohibition against producing drugs not in accordance with GMPs. 501B, prohibitions against the drug not complying with a corresponding USP-NF monograph if one exists. If there is not a monograph exists and it's a drug product that doesn't comply with the label claim, 501C applies. 501D is prohibition against mixing, packing drug products with a substance as to reduce its quality or strength. And we'll talk about that in a moment. All right, Gail, yesterday, my colleague Gail Barnell presented this slide yesterday. The take-home message from this is that from my presentation, 503As are exempt from good manufacturing practices. So during an inspection of a 503A compliant facility, the focus will, there will be no focus on GMPs. Again, it's a compliant firm. 503A facilities do remain subject to all other adulteration charges, including the prohibition against insanitary conditions. For 503Bs, they're not exempt from GMPs. They're also not exempt from any other adulteration charges, including insanitary conditions. But again, the take-home message is 503Bs remain subject to CGMP requirements. Okay, 503A facilities. So from the standpoint of the agency, there have been actions taken, advisory actions taken, and occasional enforcement actions taken to a 503, otherwise 503A compliant firm in regards to, for example, they are getting 100% patient-specific prescriptions, but they are subject to adulteration. And as indicated on the slide, to repeat, they are exempt from CGMPs. So Section 501A2B does not apply. 501A1, prohibition against presence of filth. 501A2A, prohibition against preparing, packing, or holding a drug whereby it may have been contaminated with filth. These are collectively what I'm referring to here as the filth violation. The important part, well, there's a lot of important parts within 501A2A, but it does come down to the last bullet. They don't need to be actually contaminated to be deemed adulterated. It is whether or not they've been made, prepared, packed, or held such that they might have been contaminated with filth, or may have been. All right. 501A1 is used if we're talking about a product that is intended to be sterile, considering the route of administration, and it's a confirmed sterility failure. For a non-sterile product, non-sterile products needs to at least have a limit on the number of microbes present and a prohibition against particularly pathogenic ones, considering the route of administration. We've also seen cases, inspections of both 503A and 503Bs, that there are unintended particles. We've seen cellulose fibers, we've seen wood fibers, insect parts, we've seen vial stopper residues, things chipping off and entering into some type of suspension within the liquid. So if we see that during an inspection, if we see this in product that has been distributed, then the firm is subject to a 501A1 charge. For insanitary conditions, as I indicated, if you're making a product within under insanitary conditions, there is a heightened potential that the products that being made will be contaminated, and that's the prohibition. Your facility has to be such that the potential for filth within the finished product is been minimized. All right. The best way of pointing this out is that you're talking about products that are intended to be sterile. Sterility assurance has to be built in. It cannot be tested in. If a product is intended to be sterile, and it is, and if you're making a batch of multiple units, that comes typically from a stock solution, and microbial contamination, if present, will be not uniform. So if one then takes a batch and divide it up in, for example, 100 different vials, if contamination exists, those contaminations will not be equally distributed. In addition, one of the considerations about tests is that since microbes are not, by their very name, indicates that one cannot see that with the normal human eye, there is no way, obviously, in most cases, to determine whether or not it's contaminated. So it's a situation of, if I present this to a quality control laboratory, well, this might have been contaminated. Tell me if it's not. There is no way to definitively test sterility into the product, because the chances of missing it, especially if its contamination is relatively low level, there is a huge chance of missing it. And as it turns out, failing a sterility test provides more information than a passing result. If you fail a sterility test, the product intended to be sterile, that gives you evidence that you've had some type of breach within the aseptic processing. Passing just indicates you might have missed it, with the emphasis on you might have missed it. When insanitary conditions are present, they increase the risk of drug contamination, whether we're talking about microbial contamination, whether we're talking about non-viable matters. Again, we've seen wood fiber. Chemical and unintended, potentially toxic or potent compounds. They lead to risks, increased risk in patients. And there's an article that was put out by the NIH that does look at the various pharmacy-related outbreaks over a 12-year period, in which, if you read the various case studies, all of these had some aspect of insanitary conditions present. If we talk about good sterile drug production, what the agency is looking for and is, from the standpoint of 503A and 503Bs, is for the facility to understand, or for the personnel who are working within the facility, to understand the potential sources of microbial contamination. It can be directly from the facility, from the environment, air, surfaces, water, the equipment and supplies you're bringing in from outside, drug components, drug substances, excipients, the containers, closures, and personnel. Personnel, you and I, are if we're working in a aseptic processing area, in a clean room, we are the major particle generator. There's more than 1,000 known microbial species found on healthy human skin. The number of microbial CFUs per centimeter square of healthy skin is in the tens of to 100,000. And a single healthy person present in a room can add up to 37 million CFUs per hour. Poor personal practices can obliterate aseptic conditions of the best designed and maintained facility. And this is one of the major emphasis on clean room design and personnel practices, is to minimize the impact that personnel have on aseptic processing. The purpose of gowning, you're not eliminating the generation of particles from humans. The idea is containing them and providing a barrier. When aseptically compounding drugs intended to be sterile, they must be, personnel must be free of infectious diseases, inflamed skin, makeup, and exposed jewelry. And the reason why this is that people having these conditions, or having makeup on or jewelry, is a tendency to increase the amount of particles being generated. One of the issues, for example, wearing rings on fingers, is that if you're putting a sterile glove over that, the ring itself is now presents a puncture possibility. They should be properly gowned. For 797, it says sterile gloves, clean gown, hairnets, masks, shoe covers. For CGMP requirements, and this is for the 503Bs, all outer gowning items to be sterile. The gowns must be donned such that they're not contaminated, especially the sterile gowning items. And while processing, that personnel are demonstrating proper aseptic behavior. Movements are slow and deliberate, frequent hand sanitizing, and no interfering with the unidirectional airflow of the ISO 5. FDA, the last iteration of the draft guidance, the agency is working on finalizing this. This guidance focuses on 503As and 503Bs, covers both sterile and non-sterile compounding, and it includes examples of what the agencies have seen during its inspections. This should not be considered an exhaustive list. Unfortunately, inspections come along and we see things that are highly problematic, but even in after at this point almost 8 years post NECC. The guidance is intended to provide is to indicate to compounding facilities on what constitutes insanitary conditions. The agency encourages compounding facilities to assess the conditions and practices within their facilities to determine whether or not contaminations are present, and with the recommendation that they take appropriate corrective actions as necessary. Guidance is intended to seek voluntary compliance from 503As and 503Bs to assess their facility and their practices to eliminate any insanitary conditions. And again, it also relates to the non-viables. All right. So insanitary conditions, it deals, again, if we're talking about a sterile product, the elimination of, or the prevention of microbial contamination, the contamination of non-viables. If we're talking about the non-steriles, that the amount of contamination within the product is less than the limit, and also doesn't contain the non-viables. For 501B, it refers to products. It refers to. Sorry, I have a dog who likes to interrupt me when I speak, so that was that. She was looking for something to play with. Sorry. So, Section 501B refers to the drug product in which there is a corresponding monograph. 501B, the drug must comply with the monograph quality attributes unless plainly stated how it differs. Firms should understand how to read and interpret the USP-NF because the monograph may not, if it's an injection product, it may not make direct reference to USP-1, although if it is an injectable, regardless of whether or not one is called out in the monograph, one still applies. The monograph specifications are applicable for the entire shelf life. And just to discuss very briefly, there's a difference between release specifications and shelf life specifications. If we're talking about a GMP operation, Section 211-165 is a requirement in test and release for distribution that prior to releasing a product, the batch of a product for distribution, one must determine whether or not that product at release meets specification. Shelf life specification is the quality attributes throughout the expiration date, or if we're talking about a 503A, throughout its beyond use date. And just to give you an example, if we're talking about a USP monograph and it says that the limit is the label claim plus or minus 10%, if you at release test it and it's 91%, well, it's within the monograph specification. But if you don't take into consideration the stability of the product over the entire beyond use date or the entire expiration date, you're at risk that over that period of time, the amount of the drug, the active pharmaceutical ingredient, will go below the USP monograph specification. So generally speaking, at least for a GMP manufacturer, and for the 503As need to consider at release not only if it's being tested, what the level of API is at release, but also what's the expected degradation throughout the shelf life. So as indicated before, drug quality should be intentional, predictable, and consistent. If it's a 503A firm, they're exempt from the statutory and regulatory GMP requirements, including the performance of a formal process development and validation study. But that means even though they're exempt from performance of process validation studies, then a 503A firm to be able to not distribute a product that potentially violates 501B, they need to have a set of controls that are scientifically and technically sound and robust to ensure that they can consistently meet the required specification of the corresponding monograph. All right, so if a product, compounded product, there isn't a corresponding monograph, then it's a section, then it falls under section 501C. It should not be considered to represent a looser standard. The responsibility actually then goes back onto the firm for finishing compounded drug products that have appropriate shelf life quality attribute specifications for the drug products. So they're going to have to do a little bit more work. They cannot rely upon what the USP monograph states. They're going to have to come up with their own specification. And again, similar to 501, to the products made under 501B, they need to ensure that their compounding operations are scientifically and technically sound to ensure that the finished product can consistently meet the appropriate quality for the labeled shelf life. Regarding setting your own specification, it's relatively straightforward, but it may require some discussions with the prescriber. One example, for a 501, for a USP monograph about an injectable product, it'll state the endotoxin limits. And the endotoxin limits are based upon the dose, and typically that information is obtained from FDA-approved labeling. But for 501C, compounded products, for compounded products, there isn't FDA-approved labeling. So the 503A and 503B should have a conversation with the prescriber on what the intended dose will be. And then if you look at USP-85, it gives instructions, it's bacterial endotoxin test, it gives instructions on how to set the limit. So it does require a certain amount of homework and a communications between the facility, the firm, and the prescriber. All right. In going, I mean, there are some discussions about bulk drug substances in the previous, at the fireside chat, and let me just speak about that for a while. Now, there's, in the statutory requirements of 503A and 503B, there's various references to the use of drug components that meet USP-NF specifications. Bulk drug manufacturings are statutorily required to be manufactured under GMPs. However, 21 CFR does not apply to bulk drug manufacturing, and that's stated up front. The International Standard for the Manufacture of Bulk Drug Substances, Active Pharmaceutical Ingredients, is ICH-Q7, Good Manufacturing Practice Guide for Active Pharmaceutical Ingredients. FDA is a signatory to ICH, and FDA has adopted this guidance, and it's directed towards manufacturers of APIs. If you're making an API intended for sterile drugs, the guidance indicates that water used in the final isolation and purification step should be monitored and controlled for total microbial counts, objectionable organisms, and endotoxins. And other controls are also specified to minimize total microbial counts, bioburden, objectionable organisms, and total endotoxin. Going to this, if there is a monograph for the drug product, or excuse me, drug substance, the monographs are a minimal set of standards. It does not include specifications on all quality attributes that might impact process parameters or finished drug product quality. For example, monographs rarely include specifications for polymorphs, particulate size, bioburden, or endotoxin. Now, consider this. If we're talking about sterile, I'm talking about sterile drug productions, an injectable solution. Polymorphic or particle size variation may impact the solution grade of API during mixing. From a GMP standpoint, mixing is determined, the mixing is determined through a process validation study. One qualifies the APIs, develop the specifications needed that conform to what was done during the process validation study. And then if there's a mixing time, it's determined, it is validated in a formal study. The requirements for that is 21 CFR 211-100. But for 503As, there is no GMP requirements. It's typically specified within a master formulation record. But I have spoken with 503A firms that have noted to me, off the record, that they've switched API manufacturers. And what used to work before, something that used to mix in 10 minutes, now takes 30 minutes. And I pointed out to them, you may have either a polymorph of the material, or you may have it's unusual, has a larger than expected particle size. In case anyone doesn't know what a polymorph, it's a different crystalline structure of the same substance. So depending upon how the API is manufactured, what solvents are used, the finished crystal may be different depending upon the solvent used to isolate that. If one speaks about the bioburden, for terminally sterilized product, sterilization method is validated and sterility assurance is achieved as a probability of log reduction of microbial count and is partially dependent upon the bioburden originally present. So if there is no requirement, and again for 503As, to determine the bioburden of the incoming material, there is a higher potential of having a sterility failure because the sterilization methods are built upon a certain understanding of what the microbial bioburden is originally at. So there needs to be the process, if one's using a terminal sterilization process, there needs to be an understanding of the limitations of that process. For filter sterilized product, the ability of the filter to remove viable organisms without clogging or microbial grow through, is at least partially dependent upon the bioburden originally present. And I talked earlier about the requirement in ICH 27 that talks about endotoxin. That requirement, or that recommendation really, that's the guidance, the recommendation on controlling for the quality of water is that the removal of endotoxin from a bulk component, though it is possible technically, it is highly impractical. It's far easier to start with bulk ingredients that contain low endotoxin levels. And from my position within the agency, it appears that a lot of compounding pharmacies are using APIs to make sterile drugs that were not manufactured under the conditions of ICH-Q7 that requires control of the quality of water to control for endotoxin. And to give you a case in point, this is from several years ago, where glutathione, which was complied to a USP monograph, actually the USP monograph was a dietary supplement, using it to make an injectable. The glutathione was not intended to be manufactured to make it into sterile drugs. The agency received a number of adverse event reports of what appeared to be endotoxin shock, and then when the agency tested the product, found that they did have very high endotoxin levels, which were likely the reason why all those people needed to be hospitalized. Another thing, in terms of standardization, and again, what I'm trying to have the focus is on process control during compounding, whether it's a 503A or 503B. When using bulk ingredients to make sterile drugs, it's imperative to minimize the pre-sterilization hold times to reduce microbial proliferation and prevent increases in endotoxin levels. One of the things that, and I give a reference later in the presentation, there have been a number of cases where in a prolonged, just keeping a bulk drug, non-sterilized bulk drug substance in the sterilizing apparatus for a prolonged period of time, it is been demonstrated that as proliferation occurs, the potential for the as the cells divide is for them to actually grow through or penetrate through the filter. So there is, again, needs to be an understanding of the limitations of working with a non-sterile ingredient and minimizing the hold time to ensure that you're not going to run into problems later on. For 501D, a drug is adulterated if there's any substance has been mixed or packed therewith so as to reduce its quality or strength, or substituted wholly or in part therefore. And another link to another FDA public notice, and it's something we saw a number of years ago. We do note that a number of compounding facilities are using syringes that were approved by the agency, CRH, not intended to be drug containers, but intended for administration purpose, to remove material from a vial prior to injection. The manufacturer of the syringe changed the formulation of the syringe plunger rubber tip, and that leached material into the drug products under storage, and that leach component catalyzed rapid degradation of a lot of the different types of active pharmaceutical ingredients with a significant loss of strength within minutes. And again, firms should ensure that their compounding operations are scientifically and technically sound to ensure that finished products can consistently meet the appropriate quality. And this From this is an understanding from where your drug components and container closure systems are coming from. And you may want to consider talking to your supplier and asking, to their knowledge, or has there been any indication on whether manufacturing changes have occurred? Five oh three Bs. Again, they're subject to all the federal charges prohibitive on adulteration, including GMPs. A fully GMP compliant manufacturer will not have any insanitary conditions present. A facility that does not have any insanitary conditions may not be fully compliant. They're not the same. Some of the short list of differences between 503As and 503Bs expectations, the design of facilities, the gowning, media fills, frequency of non-viable particle monitoring, extent of qualification of cleaning and disinfecting agents, extent of qualification of terminal sterilization, requirements for process validation, the requirement for release testing, and the requirement for record keeping. The agency has released draft guidance. The last one was issued just the beginning of this year, and the comment period has ended, but the agency is still reviewing comments. Just for the regulatory requirement as discussed by the preamble to 211. This is from 1978. This is how long the 211s have been around. The scope of the CGMP regulations set forth the facilities, methods, and controls used for the manufacturing, processing, packing, or holding of a drug product. And within 211, the scope, the regulation in this part contains the minimal current good manufacturing practices for the preparation of drug products. The agency understands that 503Bs, their practices and what they're intended to do, that there are differences between 503Bs and conventional drug manufacturers. So the guidance reflects FDA's intent not only to recognize, but also maintain minimal standards necessary to protect patients from the risk of contamination or otherwise substandard products. And it informs the industry and informs the stakeholders where we're going to issue, or where we're going to, how we're going to interpret the regulatory requirements as it relates to outsourcers until regulations are specified. Some of the activities were enforcement discretion, and this is outside of the temporary guidances. Drug component testing requirements of starting with FDA-approved sterile products, the expiration, dating, and stability studies, product release testing. And with that, for those who are interested and have, want to see some of the references for this presentation, this is a list of guidances and other sources of information. And with that, I will end my sharing of my presentation. Thank you.